Yes.
Okay, great. I'm Sebastian, postdoc in Berlin. I do work with a lot of data, but I'm not a machine learning expert. I work with Markov State Models on big simulation data and all these things, but since this group is more focused towards the machine learning world, I was shifting my topic a little bit. And there are actually two topics. One is about my research, and the other one is about my work with Politics Index, a nonprofit association that we recently founded. And so let's dive in directly. The reason why we're all probably sitting at home right now and having these Zoom meetings and do not meet in person is because of this beast. And I'd like to, in the next minutes, discuss with you some molecular aspects, how this virus is actually doing all this harm, and what researchers do to try to develop better vaccines or drugs against this virus with using data and machine learning techniques. And I can't give you the whole overview. I mean, it's like right now it's all about COVID-19, and everybody's trying to publish as many models as possible. But I thought I maybe just give you one particular example of what I'm doing on a related study, on a related topic, because I'm not an expert in COVID, but I have been working for many years on the adaptive immune system. And so I'd like to share some thoughts with you on this. So what you see in this cartoon, I don't know, maybe there are some people who have some biology background, maybe not. This cartoon is basically the zeroth approximation explanation of what is happening on this very complex system. And biologists usually, I'm a physicist, usually biologists start with this cartoon to have a framework that physicists then can start doing computations. And so the idea about this, what biochemists and biologists found out, is that this COVID-19 virus is very particular because it has the surface with so-called spike proteins. They almost look like a little corona, and that's why it's called the coronavirus. And so what happens is that this virus enters the host cell. I hope you can see. Can you see my mouse?
Yes, yes.
Okay, good. Just checking. Enters the host cell here via some ACE2 receptor. And the scientifically interesting aspect is now that this by itself doesn't happen alone, because these spike proteins, they have to be activated through some protein, which are basically proteins that do some chopping, and particular serines for the experts in the fields. And so a lot in biology, you have these things, one thing binds to another, activates another, and then you have probabilities of things happening. And so one big drug target that currently pharma scientists are trying to attack is actually not the virus itself, because eventually we would like to have a vaccine, of course, but at least find a way with some medication to inhibit the action of this TMPRSS2 protease, so that it would then actually close the gate for COVID-19 to enter the host cell. And so to develop these drugs, what usually happens in pharma is that it takes sometimes up to 15 years, millions, trillions of dollars. You have to find a substance that has two main criteria. First, it has to work well, and secondly, it must not kill the patient. And so you have lots of candidates here, among which you do some screening tests in the first stage, possibly also on the computer. Then you have narrowed down these molecular candidates. You move on to actual lab experiments, where you do in vitro, meaning like on cell cultures, you do experiments, or even animal experiments. that are costing a lot of money, and then in the clinical stage here when you actually work with real patients. And the goal of this pipeline, I mean, it's horrendous, especially for cancer drugs and also viral drugs. I mean, it's like insane how much money and time is being invested. And the goal is basically to try to narrow this funnel so that we can come up with better candidates, reinvest the money to get better drugs that have less side effects. And so you could either do better experiments or you could actually improve this pipeline by predicting some of the outcomes that you would have, for example, in animal experiments. And one of these tests that I've been working on is called the immune reactivity of these compounds. This could be a regular drug, or it could be even a vaccine that we can talk about, because vaccines are nothing else than proteins that are being used as a sort of drug before the patient is being sick. And so this is the idea, that you do cheap experiments on the computer that may not have 100% accuracy, of course, or any area under the curve, but at least they narrow down the number of candidates that you have to do less experiments. And to address this immunogenicity, this immune reactivity, we have to look at and potentially model how the immune system works. Because the problem is that if you have a drug and it's administered to the patient, it may work really well if there weren't just this immune system, because a lot of drugs, they don't work because they get eaten up by the adaptive immune system before they can show any effect. And this could be the case, for example, for a vaccine, or just like any drug. Let's start with this. And you have this therapeutic protein, for example, that is supposed to kill the infection of COVID-19. And in the lab, it works perfectly. But unfortunately, because of some specific circumstances of the patients, this patient develops immune reactivity towards this, which means, you know, components like T cells, B cells, and eventually antibodies eliminate these therapeutic proteins. So all the development that we have is in vain, and we can basically trash this candidate. And to avoid this, it would be nice to be able to predict this from data. Or for example, for a vaccine against COVID-19, you would actually like to have the opposite, because most of the vaccines, they're harmless to the body, but you want to make some subtle modifications so that you can actually induce an immune reaction, so that you can start the process of immunity, so that you have this entire complex cascade of T cells and B cells reacting. And so one approach of doing this is, in fact, that you are taking publicly available data about information experimentally known of particular peptide sequences, if they cause an immune reaction or not. And this is important because usually along this cascade here, what you see is these proteins, before they get recognized by T cells, B cells, antibodies, and so forth, they first get chopped down into little peptides. Peptides meaning, like, you know, you have the whole chain of amino acids that forms a protein, and then you chop this into little pieces, and then you have these small sequences of letters that you can see here that all form the protein. And this is experimentally known. So you can actually say, okay, I have like a classification. This is my peptide sequence. It causes B or D T cell reaction. Has been shown this to be the case. And then you can train a deep neural network. In this case, I employed a gated recurrent unit network because it can encounter take care of the history of the past, so that when you read these letters to get an answer, you know, adaptive T cell response or not, it looks at the context. It's basically like a word recognition, but now directly applied in a biological context. in a very simple way, because of course these proteins are not, they do not look like this. They're not simple letters, but the actual encoding, if this is done smartly, you can use these letters as a nice representation to reach your goal. And of course, I didn't do the smart encoding. I was using it from a different context. And I think this is something, yeah, I don't know, maybe something to share, the idea that a lot of times, yeah, you don't have to rebuild the network from scratch, but you can actually take a network that has been working well on a particular case and then just re-engineer it to a different problem that is related. And so what you see here is actually a neural network that predicts peptide binding. So you have these letters, and the question is, do these peptides, these small molecules, bind to MHC? And MHC is some important switch in the adaptive immune system. And the way this flow goes is basically that you include information of this peptide sequence of the protein, and then answer the question, you know, is it binding or not? So it's really like a yes/no classification. And in this case, because I would like to address immune reactivity, I took away this entire information entirely. I basically integrated over all these different aspects and just focused on the input peptide sequence for now. And developed a software where you can basically then have a protein sequence. In this case, it's an HIV virus, not SARS at this point. It chops down these sequences into little pieces, spits out some probabilities, and then I can compare this with a test data set and get some nice accuracy and AUC values that are in fact significantly better than recently published work. But this has to take with a grain of salt, because I show you here now prediction accuracies and AUC values for B cell and T cell reactions. But for particular patients, they have a very specific orchestra of T cell types and B cell types. So to be more specific and to go into personalized medicine, one then would need to basically take this whole data set of 500,000 exomes that are used entirely and basically be more specific. And that's currently the challenge, even in the field, because as soon as you want to make predictions for a single patient, you have, like, your data is set that you train and test on shrinks from this size to something very small. So this is more like a population prediction at this point. Yeah, and there are other aspects that need to be addressed here as well. So this is work in progress. This is the group I'm working in, and let me switch quickly to another topic where I'm actually using all the tools that I'm using in the research field, and now then apply them to a completely different topic, and which is political transparency. The common thing is that in both cases, I have large data sets to deal with. And every time I have to talk to experts in the field to understand the context of these data sets. So first, this was a biological problem, and I'm a physicist. I have some clue about biology, but I talk to the experts there. And the same thing here now with political transparency, the nonprofit association that we founded last year. It's the same thing, that my data analysis are only as good as the political and legal and interdisciplinary advice that I get. So we are a team of growing members, project managers, UX designers, developers, legal advisors, political scientists, Master of European Studies, marketing, that's me. And so we actually, our goal was basically to help the European Union to become more transparent and clear. And I'm making a little bit advertisement for this nonprofit right now, because we're still looking for other people to join who like the idea, because of course we're in Berlin, but we work interdisciplinary, internationally, and we would like to work both in different nations, but also all the way up to the European level. And the problem is that some of you may have seen is that the European Union is very large. It's not only one institution, but it's a ship of institutions, and the normal citizens, they might feel a little bit lost because the European Union is in Brussels, and why do I care? I mean, in somewhere in some village in Germany, and it's usually a problem because we all vote for the European Parliament. And so over the past years, the EU has started a lot of initiatives and hackathons to promote this transparency, and also, you know, checking how they're doing. And so our goal is to, like some other nonprofit associations as well, is to identify different data sources for our question. In this case, it would be a political process checking, and basically make use of all these different data containers out there, and connect them all together to answer a very particular question and come up with a tool that is useful to the citizens. And this very particular question that we already won 2 competitions with is actually this project that we started here. And it's about, it's a little bit hard to see. This is supposed to be about checking political promises. So you have a politician or a party, they say, this is in German now, we want to fight terrorism inside the country. And the different parties, they answer these questions, and you can answer these questions as well, and then you see with which party you match best. But the important thing here is actually that these parties, in even their manifestos, the election programs, they make these promises, and later you have bills in the European Parliament that are being passed, that are being voted upon, and the question is there a match? Can we take these parties that are being elected accountable for? And so there are different data sets. One is this big one, of course, which contains all the bills from the last 50 years. And we are in the process, basically I did some data science to check if this all makes sense. But the important thing is that right now we are basically having this very particular challenge that we would like to automatically screen through content of bills that have all this text here, like here, and it goes on, and make some sort of topic sentiment analysis that we would, for example, get from different data sources like this platform here, and identify based on certain keywords automatically, okay, take a look at this bill network and tell me if this is pro or against climate change, pro or against the economy and all these things. And so the challenge that we have is right now that the data is very sparse. I'm not a natural language processing expert, and it would be great if some of you would be interested and actually do have some experience in natural language processing, that we could get in touch and maybe at least get some advice how we can proceed. Because right now it seems that the only way this is maybe possible is through transfer learning from some other legal tests. Legal tests are very emotionless, and so maybe there are some other data sources and algorithms that we can transfer this from. Yeah, I wanted to keep this as brief and crisp as possible. This is the team, and thank you very much for your attention, and I'm open for questions now.